All, this is Dr. Mubin Sayed from Drbeen.com. Welcome to one more show. Today we would now start, as promised, the process of inflammation. So of course, with the SARS-CoV-2 and the COVID-19, inflammation is the primary damage that is occurring. And generally for all diseases, the final common pathway that causes or function that causes the damage is the inflammation. Inflammation's basic purpose is to actually neutralize and remove the damaging or offending substances from our body, may these be infections or other things. And then inflammation's role is also to help us repair the area where the damage has occurred. However, interestingly, many times inflammation can actually do, or the response of our body that causes inflammation can do more damage to our own tissue than the offending substances. That is what then causes the disease and symptoms. And many times when inflammation occurs, for example, let's say that my skin has inflammation, many times when there is a repair done, the repaired tissue is mostly fibrosis. Original tissue is very rarely reproduced. There are conditions for the original tissue to be reproduced. Most of the time after the damage and inflammation, the reparative area, the repaired area is scar tissue, and scar tissue is non-functional. So on one end, inflammation is very useful. On the other end, it can be harmful. And then inflammation can become chronic as well. In some people, it just continues for a very long time. And that is where the autoimmune diseases come in. And that is where these issues where somebody has their own body attacking their own tissues and causing continuous inflammation forever. So I wanted to start this discussion today. Again, the context is SARS-CoV-2 and COVID-19, but the idea is to understand what really happens in our body more than just the immune system. So let's start our discussion. I'm going to share my screen. So a few links that I have in the description. This is a study about the Fc portion of the immunoglobulin and the receptors. It will come up in the discussion in a bit, but this is a link to that. This is a Wikipedia link about Azurophilic granules. These are the granules present in neutrophils that help with the combating the pathogen, and then they can cause inflammation as well. Then this is a link to NADPH oxidase, which is an enzyme present in the neutrophils which take part in the clearance of the pathogen and then can also cause inflammation and tissue damage. Then this is another article over here, which is about the antiviral innate immune response in non-myeloid cells augmented by chloride ions. The point of this study is that when we would study the inflammation process in a bit, you will see that finally what we do is whenever we catch a bacteria or a fungus or a virus or a dead tissue of our own body or an old cell in our own body, eventually we break it down by burning it. And that burning is done through bleach. So this is that article that talks about how hypochlorous acid, which is an active ingredient in bleach, how that is formed in our cells and how that works. Then here is the process of halogenation. And halogenation is another process that helps to break down pathogens, for example, coronavirus. So we'll come across that in our discussion too. And then this is the process of lipid peroxidation, which is another way that we damage the pathogens. So all the links are here. Let us now start our discussion. This is actually a fascinating area, and I am super excited to be talking about it today. So let's start from here. This is Nina Kant from Twitter, and she sent me this cool bean friend. So I like this cool bean idea over here. Okay, so inflammation. Inflammation is a protective mechanism. The function of the inflammation is to remove the initial cause of cell injury. So whatever is going to cause a cell injury, and please remember, inflammation can remove some causes of cell injury and it cannot remove another. For example, if somebody is under a radiation, or if you are in sunbathing and the radiation is causing damage, and you don't get up and go in sun, or you don't have a sunscreen, then the inflammatory process in our body cannot remove the sunlight. So there are some things it can control and some things it cannot control. So first, it would try, the process of inflammation would try to remove the offending agent, and then it would try to repair the tissue that has become damaged. How does it try to remove the offending agent? Let's say we talk about coronavirus. The inflammatory process is going to try to dilute the virus. So by creating lots of fluids around it and kind of diluting it in there, or it would try to destroy it, which we have been seeing earlier as well, and we'll see today as well, or it would try to neutralize it. What we do is we kind of pack the pathogen with lots of complement and immunoglobulin. We kind of wrap it in with multiple proteins and kind of remove its dangerous effects. So that is the process of neutralization. And finally, inflammation is also responsible to trigger the process of repair, which may repair the actual tissue, or most of the time it will result in scar tissue. So that is inflammation. The problem with inflammation is it is a healthy mechanism. It is a mechanism that helps us all the time. The problem is that sometimes healthy tissue becomes damaged as well. And that is where the bad effects of inflammation come in. So Pia Dagani says, it is very late here in Italy, so I have to wake up to get connected with you. Thank you very much. Continuing on with the discussion, the participant of the inflammation, what things take part in the inflammation? So there are cells. Some of these cells we have already heard of, for example, macrophages, dendritic cells, neutrophils, T cells, B cells, they are the cells. So immune cells take part in inflammation. Endothelial cells, or the surface cells of blood vessels and other tissues as well, can take part in inflammation. And then the cells of the connective tissue, that is the normal tissue that is present in our body, called connective tissue, that connects various cells together. These cells can take part in inflammation as well. In addition to that, there are molecules, there are chemical substances which also take part in inflammation. So inflammation is a process run by cells and proteins. And the proteins can be plasma proteins, and then other substances can be oxygen. Of course, oxygen is not a protein, or chloride or nitric oxide and other substances, different molecules. So cells and molecules come together to produce inflammation. Inflammation itself is of two types, acute and chronic. Acute means immediate, short-term, and transient. Chronic means something that is going to live longer. So, for example, if I become infected with coronavirus today and my body responds to that, we will call that an acute inflammation. But if that response continues over months and years or weeks, then it would become chronic. Most of the time, in the acute inflammation phase, for example, if some part of our skin gets damaged, what happens is there are vascular changes. So you may have noticed that when we get injured, that area becomes red and swollen and painful. So it becomes swollen because vessels become permeable and the fluids ooze out. So vascular changes occur, and then cellular events occur. For example, neutrophil would arrive in that space, in that infected area or damaged area, and try to take care of the area and remove the offending particle. In case of chronic inflammation, that is when inflammation continues for months and years. For some people, they continues for their whole life. In that case, immune cells do participate, but most of the time in this chronic inflammation, macrophages, T cell and B cells take part more than the neutrophils. So chronic inflammation, and maybe for the SARS-CoV-2, as we know that there is macrophage activation syndrome, and then we also know that there are long haulers. So it is possible that the acute inflammation triggers macrophage system, which when becomes activated, then that becomes chronic inflammation. Tissue destruction occurs in chronic inflammation. And then there is repair process that just continues. So in chronic inflammation, damage continues to occur, repair continues to occur, and the tissue that is becoming continuously damaged and repaired slowly becomes scarred and becomes useless or functionless. For example, liver cirrhosis. When in the liver cirrhosis, there is continuous damage to the liver, and then repair is by the scar tissue. And over decades, that ends up with the liver that doesn't have functional units in it, and it has more scar tissue in it. So, and that repair is done by fibrosis. Fibrosis is the scar formation and new blood vessel formation. So what is important here to take away is that in acute cases, it is blood vessel permeability and neutrophils that are more active. In chronic cases, it is macrophages, T cells, B cells, other immune cells, plus continuous damage and repair process, which causes chronic inflammation. Now, acute inflammation, again, context SARS-CoV-2, trauma, for example, blunt trauma or sharp trauma, or trauma of any kind, accidents, infections, foreign bodies, for example, splinters, immune reactions, for example, allergies, and then autoimmune disorders. Now, remember, allergies and autoimmune disorders can actually become chronic inflammation as well. But there are some hypersensitivity reactions that are acute. They occur at that time. For example, some wrong drug injected in a person and an acute inflammatory response occurs. That is an allergic response, but that is acute. And if we can manage the patient and save them, it is not necessary that that response would stay forever. On the other hand, there can be allergic reactions or there can be autoimmune disorders that can continue for a long time. For example, asthma is one example of that. So hopefully this much is clear, that what is inflammation, what are the types of inflammation, what are the kind of cells that take part in inflammation in both types. And we would do chronic inflammation separately. Today we are going to focus on acute inflammation. So in the acute inflammation, first of all, what happens is there are vascular changes. We just talked about it. And I have deliberately not done the vascular changes today. Today, I want to focus on phagocytosis. Tomorrow, we'll talk about the vascular changes and how the cells reach that place. So it is, you can say that it is kind of upside down, that we should have talked about the cells reaching the area of infection before we talked about the cellular function. But I thought that we have already talked about immune systems structure and function a lot. So this phagocytosis is very attached to the mechanisms that we have seen before. So it made sense to talk about phagocytosis first and then talk about how the cells arrive in that place. But vascular changes occur in acute inflammation. Leukocyte recruitment occur. Recruitment means leukocyte, white blood cells that are running around in our body, in our blood, all over the places. If, let's say, I have an injury in my hand, then we would try to recruit white blood cells in that area. So the blood vessels in that area would allow or enable, plus the chemical substances in that area, would enable the white blood cells to come out of the blood vessels in the area of injury and start helping there. So that is called leukocyte recruitment. And finally, leukocyte activation occurs. And this is the thing that we are going to talk about today. Phagocytosis is a part of an activated leukocyte. So leukocyte or white blood cell activation means what? What happens is and we're going to look at that diagram in a second. What happens is that white blood cells will come, for example, in this case, neutrophils. There are neutrophils, basophils, eosinophils. Then there are other cells as well, T cells, B cells. Today, we are specifically talking about neutrophils. And neutrophils are part of the innate arm of the immune system. So engagement with phagocytosis. So let's say somebody is infected with a virus. Wherever that virus is, neutrophils will come and they will try to engage with that virus, number one. Then what they would do is they will eat up that virus or pathogen, whatever that is, virus, bacteria, fungus, and they would eat it up, take it inside, and then try to digest and kill it. They have tiny stomachs in them in which they would bring the pathogen and then digest and break it. And finally, neutrophils would also, or acute inflammatory system would also then trigger the repair process. So what has it done? It has done three things: arrived in the area and engaged with the pathogen, cleared the pathogen in the area, and then tried to start the repair process. Now, it is not necessary that always this cascade of things occur successfully. And when it does not occur successfully, then our tissue gets damaged. So now let's talk about the neutrophil activation. That when a neutrophil arrives in an area where there is an infection, how is it activated? So let's start from this side. This is a neutrophil here. These things, these rock-like things, is the nucleus. It is a polymorphonuclear cell. That means the nucleus has multiple lobes to it. So the structure of the nucleus is not just one rounded thing. So it is a polymorphic nucleus. So here is the nucleus. There are some receptors on the surface. We'll talk about that in a second. These receptors are for the engagement with the pathogen, or to eat up the pathogen, or to cause phagocytosis. Then there are these little vacuoles here. Vacuoles are small bags, or you can call them lysosomes, or they are stomachs of the cell. And when we'll bring the pathogen inside, these stomachs will be engaged with the pathogen, and these stomachs will have enzymes to break down the pathogen. So these are called lysosomes, and the neutrophils' lysosomes are called Azurophilic granules, because they, under the microscope, when they are stained with the right stain, they look like burgundy color, or they look azurophilic. So that is why they're called azurophilic. Generally, they can be called lysosomes. Now, look at this one, starting from here. So this is called a serpentine or G protein receptor. So if you see here, it has these molecules, or the protein that is transmembranous. Transmembranous protein means that it is outside, plus it spans the membrane and goes to the inside as well. And there are 7 units, 1, 2, 3, 4, 5, 6, 7. These 7 proteins are then chained together like a snake. So some parts are outside the membrane and some parts are inside. So imagine a lake, a surface of a lake, and imagine the lake, Loch Ness Lake monster. Some parts are out and some parts are in. And if there are 7 parts, then that is a G protein. So this is that G protein. I always think it looks like a snake, and that is why I always make a snake head with it. In the actual cells, they don't have snake heads. So these proteins have receptors on them that allow the neutrophils to travel towards the area of infection. We will not talk about how that travel occurs today. We'll talk about that tomorrow. That is the process of diapedesis and chemotaxis. We'll talk about that tomorrow. Today, just keep in mind that there are receptors on the cell surfaces that engage with the chemotactic factors, and then the cell goes in that direction. How does it go? We'll talk tomorrow. Now, other receptors, keep this in mind. So look at this. This is a tall-like receptor. This is a receptor on the surface of the neutrophil. And what happens is, this receptor would allow the neutrophil to bind with the pathogens. So if this is a pathogen, this tall-like receptor would help the neutrophil to attach with the pathogen. Similarly, it can also attach with other receptors as well. For example, there are receptors for Fc portion of immunoglobulin, and I'll talk about that in a second. There are also receptors for complement system, and that would also something that we'll talk about in a second. Then there are other receptors too. For example, here is a receptor for interferon gamma that would activate the neutrophil. All of these receptors eventually allow a neutrophil to become ready to eat up a pathogen and to digest it. This is called neutrophil or leukocyte activation. So activation is a process where the neutrophil would engage with some pathogen, and then as a result of engagement, it would become active and get ready to digest it and break it down by oxidative burst, or by burning it, or by pouring bleach on it. So that is a process. What kind of enzymes are present in the digestive system? Just like we have a stomach, right? And our stomach has some acids in them. Similarly, this stomach of the neutrophil that is actually going to then break down the pathogen, it has many kind of enzymes in it that would be helpful to burn the pathogen, kill it, and then digest it. So what are these things? For example, there are defensins. Defensins are proteins that would break down the pathogen that is inside the neutrophil. There are myeloperoxidases. These are enzymes. We'll talk about them in a second. Phospholipases, acid hydrolases, for example, elastase, permeability increasing protein. There are proteins in the stomach of the neutrophil that would allow the pathogen to develop holes in it and make the pathogen more permeable, which would allow the fluids to ooze in and out of the pathogen and kill it. Very interesting. Lysosomal enzymes, cathepsins, and so on. So there are multiple kinds of enzymes. The takeaway here is when a neutrophil will become engaged with a dead tissue part or with a pathogen, it will become active, and for that it has lots of surveillance mechanisms on its surface that would allow it to engage with the pathogen and become active. And when it becomes active, it has in its arsenal, it has lots of weapons to kill that pathogen and digest it. So now let's see how does that happen. So the first part of the phagocytosis is called opsinization. Opsinization is the process in which we take a pathogen and we make it sweet. So our neutrophils have a sweet tooth. They love to eat sweet things, candies. So what we do is we take a pathogen or a bacteria or a fungus or a virus or a broken tissue cell, and we slap on a bunch of candies on it. And that would make it sweet for the macrophages and neutrophils and other things to eat that. So if you see here, let's say these are coronaviruses. There are many ways to opsonize them. Opsonization means to make a pathogen sweet for us. So when you bake a cake and if you put sugar in it, that would make it sweet. So adding the sugar to the cake so that it becomes yummy for us to eat, that adding the sugar part is called opsonization. So when we attach things to a pathogen so that it becomes easier for the immune cells to eat that pathogen, then we say we have opsonized the pathogen, and the process is called opsonization. So here, if you look at it, this is a coronavirus. We have attached lectins with it. And remember, we have done a talk about galactins. So lectins are small molecules that can connect with the sugar part of the pathogen. So here, if coronavirus has some sugar areas, glycoproteins on its surface, lectins that we produce, these are molecules, these molecules would then coat the coronavirus. And when they would coat it, that coronavirus would now become yummy. And what happens is, if you see here, this is a neutrophil, and neutrophil has a receptor called C1q. And you may remember C1q from our complement discussion. So here is a receptor called C1q. This receptor would bind with the lectin, and lectin that is connected with a pathogen. So in this way, the receptor or the neutrophil would be able to attach with the pathogen. Remember, ACE2 enzyme is the preference of the coronavirus to connect with us, with our cells. On the other hand, we also have ways to connect with coronavirus. The only difference is when coronavirus connects with ACE2 enzyme, then it comes into our cell and causes damage. When it connects with these receptors that I'm showing right now, then we damage this by bringing it directly into our stomachs, or cells' stomachs, and killing it. So one binding mechanism is we attach lectin to the virus or bacteria, and then we have receptor for lectins on our neutrophils. When this binding occurs, then the virus will be taken in the cell. Similarly, here is the virus, coronavirus, and let's say we have made antibodies, those famous antibodies, binding antibodies and neutralizing antibodies. So we made antibodies, our plasma cells had become active, antibodies have been produced, and here is antibody bound to the coronavirus. The one part of the antibody is called constant region. that constant region of the antibody can connect with neutrophils and other phagocytes, because phagocytes have a receptor on them, which is called Fc gamma receptor, or receptor for immunoglobulin G. So when immunoglobulin G attaches with the pathogen, then what the cell does is the other part, the cell has a receptor to bind with immunoglobulin G and then internalize the whole complex. This is also, I am so happy that we're talking about these things. I love talking about these things. This is also the mechanism that is that causes antibody dependent enhancement. Remember that we talked about it a couple of days ago. When the antibodies are produced by vaccination or by a weaker presence of the virus or by a cross reaction, it is possible that as soon as the virus comes in, the antibodies are already present and they connect with the virus. On the other end, they connect with the neutrophils or macrophages, and now the virus gets internalized and virus finds another mechanism to get into the cell. If these antibodies were not present, virus would need to connect with ACE2 and get into the cell. But now we have given it one more vehicle to go into the cell, and that is called antibody-dependent enhancement. So here, this is one more mechanism that we can opsonize. And how do we opsonize here? We have connected the virus with immunoglobulin G, and then we take it into the cell. Then here is one more mechanism of opsonization and then eating the virus. And what is that? We connect the virus with complement proteins. Remember a few days ago, we talked about complement, that when a virus arrives in our body, our complement system becomes active, and it has many ways to become active. Eventually, it creates C3b, complement part. It's a protein. That C3b also coats the virus. It is also candy. It also gets attached to the virus. And then the neutrophils and other phagocytes, they have receptors for C3b. So this is the sweet tooth. They have a tooth for C3b. So when the C3b is attached to the virus, then this receptor can engage and the virus can be brought in. So as you can see, there are multiple processes before phagocytosis can occur. All of those processes are trying to coat the virus with something that to which our cells can then connect and capture it. So this is like if you're trying to pick up a coal in a fireplace and you have a little thing, iron thing with which you capture it. That is the same thing. So if the virus is the coal here, we have antibody, or we have C3b, or we have lectin. And once we attach those to this coal, then we have receptor for these things that would then attach that little rod and then bring the virus in. Good? So far, so good? Any issues so far? So there is a question. Guy Telfer says these are all polarization mechanism. Polarization mechanism, or really the mechanism for which we call them opsonization. These are the mechanisms that make a virus or a bacteria or a fungus that may be slippery. They make it easier for our immune cells to eat them up. So let's say if this is a pathogen and this is very slippery, an immune cell cannot really grab it, then what we do is we make it rough by attaching C3bs on it, or lectins on it, or antibodies. And when that happens, the surface becomes rough, and now the immune cell can bite it and eat it. So that is the process of opsonization. Now let's see, once a pathogen has become opsonized, how do we actually eat it and destroy it? So what happens is, imagine this is a neutrophil. We start from here. So the neutrophil, here it is connected, the pathogen, to the neutrophil, and that connection is by immunoglobulin receptor, or C3b receptor, or lectin receptor, or some other receptor like Toll-like receptor, many receptors, or there is also called PAMs, which are also receptors for this. So once the engagement occurs, then what happens is our cell, for example, neutrophil, it creates pseudopods. What are pseudopods? Are this way. So if you think about it, if I am a neutrophil, So I'm going to make my screen a little bit bigger. So let's say if I am a neutrophil and I want to eat this pathogen. So instead of bringing the pathogen in my mouth, what I would do is, imagine this is the neutrophil. It would create a pseudopod. What that means is it would create extension like this, a rounded extension, and engulf this pathogen. Then that extension, the whole thing, this bag that is formed, will be brought in. That is how the phagocytosis occurs. So if I go back to the screen now, here we connected, then we started surrounding that pathogen with the cell's membrane. These are called pseudopods. This really should not be called a pod. These should be called a pseudopocket, because it's a three-dimensional pocket structure instead of pod, which is just like a hand or an arm. But anyways, we surround the pathogen, and then we bring the pathogen inside by engulfing it in the pocket. Now this is called a phagosome. This is phagocytosis. We have eaten up the pathogen. Then what happens is these azurophilic granules, or these are called lysosomes, which are rich, they are stomachs of the cell, and they're rich in burning acids and enzymes that are going to break down this virus. So what happens is we engage a phagosome, which is the one that contains the pathogen, and a lysosome. We fuse them together. So we bring the stomach near the food, and we allow the food containing pocket to fuse with it. This is called phagosome lysosome fusion, or phagolysosome formation. In many people, there are sometimes, not many people, in some people, there are issues with the movement of the particles. For example, Shadiak-Higashi syndrome. In that syndrome, what happens is intracellular transport of material. So if you sometimes look inside the cell, one cell is a complete universe, and there are highways there, and there are tubular structures there, and there are tractors and trolleys and cars, and there are things moving around on the highways. So that is called trafficking system within the cell. These trafficking system or microtubular systems are necessary for these things to move from the cell surface to come inside, and then from inside to come near each other and fuse. In Shadiak-Higashi syndrome, for example, the trafficking system or the highway system is not functional. So those folks have cells that do not have roads in them. So this fusion does not occur. So even when they can make a phagosome, even when their cells can eat a pathogen, they cannot fuse them with the stomach because the trafficking is not occurring, and so they develop diseases. So in normal healthy people, the phagosome and lysosome will be fused together, which will cause the enzymes present here in the stomach to be poured on the virus or pathogen. So the pathogen was thinking that we are offering that pathogen a hot tub. There is a nice, beautiful, warm tub, and he's swimming in there. And all of a sudden, we're going to start pouring bleach and acids and enzymes to kill it. And then the pathogen would know what happened. So here, now the pathogen is dead and broken down. How does that happen? So let's come here. So now imagine that this unit is what we are looking at here. We have expanded it. So here is a little pathogen, which is in trouble now. You can see from his expression, he's not very happy. So what happens is, inside the phagolysosome, there is an enzyme called NADPH oxidase. This enzyme is very interesting. What happens is, it binds to the membrane of the stomach, and it faces inward, and then it causes its action. It is a digestive enzyme. If it does not bind to the membrane, so like this, binds to the membrane, and this end of this enzyme is digestive. If it doesn't do it, then if it is roaming around free, then it would actually break the digestive stomach pocket itself. Just like we develop ulcers in our stomachs where our acids damage our own stomach lining, similarly, NADPH oxidase, if it is not bound to the membrane, then it can actually damage the stomach and break it down. So normally what happens is NADPH oxidase, that enzyme will come, connect with the membrane of the phagolysosome, become active. And when it becomes active, what it does is it takes NADPH, and we have talked about NADPH before as well, and it takes oxygen. So oxygen is necessary and glucose is necessary. A cell would need a lot of glucose and oxygen to burn the pathogens. So it takes NADPH plus oxygen, it converts that to NADP, and then it produces reactive oxygen species. These reactive oxygen species, for example, here, superoxides, can attack the pathogen and kill it. Superoxides can also make H2O2, which can also attack the pathogen and kill it. H2O2 can also be converted in the presence of an enzyme called myeloperoxidase. This enzyme is going to take chloride, which is a halogenated ion or halogen ion. It takes H2O2 plus chloride and makes HOCl or hypochlorous acid. This is the main ingredient of bleach. This bleach, when we pour it onto the pathogen, it destroys it. It causes reactive activity inside the pathogen. It destroys its lipids, it destroys its other structures, proteins, and it just kills it. So there are three reactive oxygens. Reactive nitrogen species can be produced as well, which would do the same function. So oxygen, H2O2, HOCl or bleach, all of those would attack this pathogen and they would kill it. So once the pathogen is killed, now we need to dismember it. Now we need to chop, break it down. And how do we break it down? Remember, we saw the enzymes up here. These enzymes, for example, acid hydrolases, elastases, permeability increasing proteins, lysosomal enzymes, cathepsins. So the most important here, keep in mind, is elastase. These enzymes are then going to go into this cell, attack this pathogen, which is dead now, and then break it down into smaller pieces. So now the pathogen is really digested as well. Now, remember, we have also been talking that the immune cells, for example, macrophage, will take a pathogen, break it down, and then present it on its surface to naive T cells. This is the breakdown process. So once we know that this is the process of breakdown, those tiny pieces that are produced by dismembering the pathogen, those pieces can then be displayed on the surface of the cell to then make other cells aware that there is a pathogen, there's an enemy who looks like this. So what are the actions that happened in that stomach of the cell? Oxygen burst occurred. Oxygen burst is when we take a lot of oxygen, we take glucose, then we use NADPH oxidase, and we create bleach plus reactive oxygen species. This is called an oxygen burst, or fire is produced. Halogenation occurs. The chloride part in HOCl is the halogenated part, which can cause severe damage to pathogens. You know that if bleach is, if it is applied on our surfaces, it would cause damage. If it is applied on our clothes, it causes damage. So it's very, very damaging. So our body's mechanism to kill pathogens is to put bleach on them. So that is called halogenation. And then lipid peroxidation or protein peroxidation occurs as well. So there's a question, Rosemary says, does this cause natural immunity? Of course. So this is part of the innate immune system. So these cells like neutrophil or macrophages or monocytes, they do not need to be ready. As soon as they see a dead necrotic tissue or they see a virus or a pathogen, they would, excuse me, they will pick it up and they would do this process of phagocytosis and killing it. So yes, it is innate immunity. So what happens now? These reactive oxygen species and nitric oxide and the HOCl, what do we do with them? They are very damaging. And so they've done their function of damaging the pathogen. Now they can damage us as well. So what happens is, luckily, we have certain enzymes present that can destroy these reactive oxygen species. So for example, we have superoxide dismutase, that is an enzyme called SOD. SOD enzyme takes up these oxygen radical and converts them to 2H2O2 or H2O2s. Then H2O2 can also be acted upon by another enzyme called catalase, and catalase would convert that into more innocent and harmless water and oxygen. So eventually we have mechanisms to take care of the reactive oxygen species that are forming, but when we become sick and there is a lots of reactive oxygen species being produced, that would then come out and start causing damage to the surrounding tissues. That is why it is important to take vitamin C's and other reactive oxygen species neutralizing agents like CoQ10. Okay, so far, are we comfortable that we know how the pathogen was engaged, how it was opsonized, made yummy, brought in, attached with the stomach inside the cell, then the stomach had enzymes and acids and the reactive oxygen species that attacked it and killed it and dismembered it. Now, let's see how our tissue becomes damaged in this whole process. So, so there is a comment by Machiavelli, interesting, our own body creates bleach. Yes, our body's final mechanism to kill pathogens is bleach. Pathogen, excuse me, pathogens or necrotic tissue or anything they have taken up to destroy, they use bleach to finally destroy it. All right, so here, now let's see how the damage occurs. Number one here, this is a process called regurgitation. What happens in this process is that my apologies. So I need water. Give me 1 second. Sorry about that. So what happens is, in the process of regurgitation, imagine that we have the phagosome forming here. And then before the phagosome becomes a complete phagosome, becomes a complete pouch, it actually, the stomach of the cell becomes connected right away. I'm okay. I think it's just that there was so much smoke in the valley because of the fires, which is now subsiding. But sometimes that chokes me up. So this phagosome is not yet completely closed. It is still open on the outer side, and the stomach has engaged already. So when the stomach already engages, the enzymes inside the stomach fuse with the phagosome and they start leaking out. So when they leak out, they go into the tissue, surrounding tissue, and they start causing digestion of the tissue and breaking it down. So that is one mechanism in which the inflammatory process, which is trying to clear the pathogen, starts causing damage to the tissue itself. Then, so this is called regurgitation while feeding. So when the neutrophil is feeding, it is regurgitating the material out as well, because the stomach has connected with the building phagosome already has a stomach connected with it. And now the acids from the stomach and the enzymes are now going outside. And they're going to cause the issue. So that is regurgitation. Then there is another process called frustrated phagocytosis. Frustrated phagocytosis is, for example, let's say this is our kidneys glomerular system or kidney tissue, or it can be our respiratory tissue. Some tissue on which there is an antibody. So let's say this is an autoimmune disease against the renal system, kidney system. So in that autoimmune disease, there will be antibodies that will be directed to the surface of the kidney or the kidney's tissue. So when they are connected there and the neutrophil comes in, or macrophage or other phagocytes, they come in and they try to eat that antibody and whatever is attached with the antibody. So this is like we trying to eat a truck, or we trying to eat a mountain. So we cannot eat that even if we try to bite it. So now the cell has bitten here, and it cannot really eat that big chunk of tissue, and the stomach has become engaged, and the acids and the enzymes from the stomach are going to leak out. Where are they going to leak out? They are going to leak out on the tissue where the antibody was attached. And now they're going to cause damage to the tissue. For example, this is how glomerulonephritis occurs, or this is how acute respiratory distress syndrome occurs in coronavirus. So this is called frustrated phagocytosis. And it is one more mechanism where the cell, so look, this cell looks very worried. Why it is worried? Because what it has ingested are some harmful substances. For example, urate crystals. These crystals, when they come in, they would actually puncture the phagosome, lysosome. They will puncture the stomach. This is like we eating blades, for example. And so that is going to go and cause the phagosome, lysosome to break down and puncture. That punctured system is then going to cause the cell to become damaged, and the cell is going to die. And then cell would leak out, and then the inflammation would start, and then damage would occur. So this is one more mechanism in which inflammatory damage occurs to our own tissue. Now, here are some diseases that are caused by acute inflammatory system going wrong. Acute respiratory distress syndrome, where our body is trying to, our neutrophils, our macrophages, our immune system is trying to react to the pathogen, and in that process, it has done one or two, or there is a fourth mechanism as well, where the cells release cytokines. They regurge their chemical substances by intention in the local tissue, and that causes damage too. So ARDS is an example of inflammatory damage. Transplant rejection, acute transplant rejection. Somebody got a kidney replacement, and that transplant died within a few minutes or hours. That is acute transplant rejection, and that is an example of acute inflammation. Asthma, eosinophilic asthma attack is acute inflammation. Glomerulonephritis can be acute inflammation that can then become chronic as well. Septic shock is acute inflammation. Vasculitis is acute inflammation. So these are the examples of the damages that are done. And finally, some defects in the neutrophils that make their function not correct, and we cannot clear out the pathogens. For example, leukocyte adhesion deficiency. What that means is leukocyte have to come to the area of infection and attack there, correct? For that process, they have to come out of the blood vessels. And to come out of the blood vessels, they have to adhere to the surface of the blood vessel and then come out of it. And sometimes they cannot adhere. They have genetic deficiency in them to adhere to the blood vessel walls. So now leukocytes are present. Infection is there. Blood vessels are permeable, but still the leukocyte cannot bind to get out. So that is leukocyte adhesion deficiency. It's of two types. We'll talk about this one when we'll talk about the leukocyte migration tomorrow. Then there is chronic granulomatous disease. So what happens in this case is that, so let's say here is a leukocyte, neutrophil. There are pathogens. Neutrophil has eaten them, and they are in the phagosome, lysosome. They are inside the stomach. But the neutrophil doesn't have the enzymes for the oxygen burst. For example, NADPH oxidase or called phagocyte oxidase. That enzyme that puts the fire on by creating this oxygen radical, that enzyme is not working. So we have brought the meat that is a pathogen or the vegetables, but we cannot cook them. We cannot have fire to cook them. And so now the pathogens are eaten, but they're still there. So they'll just keep surrounding and multiplying, and they'll keep increasing in number. That would then cause our macrophages to become upset with this situation, and they will come and they will make a wall around this whole thing, this mess. This is like a quarantine. Just like nowadays we are quarantined in our homes. Similarly, because this area infection, our body cannot handle this infection, what happens is our body's response is to create a big lockdown in that area. So we create a quarantine, and the macrophages come in and they make a pocket in there, and they surround it all and they lock it down. This is called a granuloma. So when the digestive enzymes are not functioning correctly in the neutrophils, we cannot kill the pathogens correctly. Then those areas start becoming quarantined in the form of granulomas, and the person has granulomas all over their body. Wherever infection is, granuloma forms. Then I talked about the Shediac-Higashi syndrome as well. In this syndrome, what happens is that the trafficking system inside the neutrophils or other cells also is not working correctly. So we can form the phagosome, but we cannot connect it with lysosome. So phagosome is present, lysosome is present, but they cannot connect together. They cannot kill the pathogen. And so the infections continue to spread. So that is the discussion for today. My apologies that I had that little, I choked up in the middle of the session, but I hope that this is clear. This is called phagocytosis. Please do me a favor, like, subscribe, and share. And tomorrow we'll talk about how do the cells, the neutrophils and macrophages, how do they end up in that place? What is the chemotactic factor? How do they work with the chemotactic factors and reach there? This is also part of the inflammation. So the first part of the inflammation is for the cells to arrive. Second part is to do phagocytosis, as we saw today. Third part is to activate the T cells and B cells. Fourth part is to then cause repair. So we know the T cell, B cell things already. We now know phagocytosis as well. Tomorrow we'll talk about arrival of the cells. So with this, thank you very much for today, and I would see you tomorrow.